share the screen. So let me tell you a couple of things about myself. So I am a physicist by training, a theoretical physicist. I did my PhD in statistical physics, phase transitions, renormalization group, and things like that. And then when I was doing my postdoc, I went to a meeting of the American Physical Society, and I heard John Hopfield talking about Hopfield models of neural networks and how things that were familiar to me from the spin class literature could be used in some sense to model how the brain stores memories and how the brain retrieves those memories. So I was completely hooked. I was told by my advisor, who was a very famous physicist, that this was not physics, but I decided this is what I wanted to do. So I moved into neural networks. I worked for many years at Bell Labs. I had a very powerful neural networks group, but I became more and more interested in how the brain does it. You know, we would have invited talks by neuroscientists because we hope to incorporate some of the features of how the brain processes information in our neural networks. And I became more and more interested in understanding the brain. So now I consider myself to be a theoretical and computational neuroscientist. But what I wanted to talk to you today about is to how some of these methods that you have been learning about during the course can be used to analyze data in a real life setting. And because I am a neuroscientist, I will show you neural data as an application. I will use neural data to illustrate how we go through the pipeline and how we analyze all the data. And in the meantime, through the talk, I hope to convince you that it's very useful to learn linear algebra. It might look to you like it's a very boring discipline from the, I'm going to say 19th century, but of course it goes much earlier. And to me, it's amazing how I continue to learn about new features, new tools within neural algebra, and how it conceptually helps me, even when I'm facing nonlinear problems, to start with a linear approach and to try to understand how things work in the linear case. So I hope that this is one of the messages that I leave with you today, is that always start linear and learn your tools for linear algebra and linear methods. Okay, so this is the kind of things that I don't do, but my collaborators do. They do experiments with subjects, sometimes humans, sometimes monkeys. And let me give you a very, very simple example, which is a little bit like the harmonic oscillator in people who study motor control, because it's a very, very simple task. It reaches to 8 targets that are circularly and symmetrically and isotropically distributed about the center. So this is the task. I ask you, maybe if you are doing it, I'll ask you to touch the screen. I'll ask you to touch the screen at the center, and one of the 8 targets will come on, will be lit. There will be a waiting period, a delay. It's called an instructed delay, because you don't know how long it's going to be. It's going to be variable from trial to trial, and it's between half a second and a second. And at that point, you'll get a go signal. When you get the go signal, you start moving. You move from the center to the target that you're supposed to reach on that particular trial. You hold there and you get a reward. If you are a monkey, you get a lot of sugary beverages, which I'm sure not good for your teeth or your diabetes, but that's what monkeys seem to like, and that's what they get. So it's a center out task. You go from the center to the outer target and come back, and you get rewarded. And then the next trial involves a different target, and the targets are chosen randomly from trial to trial. So the data looks like this. You are doing the reaches to the targets. I'm going to use a color code for the targets. And what we do is we measure neural activity. I'll show you how we measure neural activity. We measure neural activity in an area of the brain which is called your primary motor cortex. Your primary motor cortex goes like kind of from earphone to earphone. And we capture of the order of 100 neurons. And this is about 20 seconds of data. I'll show you in more detail what this data is like. Every row here corresponds to a different neuron. And the ticks are action potentials. You know, the neurons communicate by emitting spikes that last only one millisecond or less. And they go beep, beep. And actually, if a neuron is very busy, like this one here, and you put an electrode on that neuron, you hear purr, 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 because its action potentials are coming very close after each other. So this is how the neural recordings work. So as I said, this is your primary motor cortex going kind of from ear to ear. And along the primary motor cortex, you have what is called a homunculus. You have a map of your body. And we know that because we can put a very, very thin wire and inject in that area of your brain a very tiny current, and we see what part of your body twitches. So if what twitches is this finger, then I am in this finger area of your primary motor cortex. So because we know this map, now look, it's a very interesting map because it's a whole body, but it's not proportional really. For instance, there is a big amount of real estate devoted to your hands, as you would expect, because we use our hands for very dexterous, and we need to have a lot of motor control to separate fingers, this and that. But what's surprising to me the first time I saw this was how big the feet are. It turns out that you really need the feet to run through the savannah, to be stable and to escape the cheetahs that are trying to get you and this and that. So you also have a lot of real estate devoted to the feet. You have, of course, face, and within the face, the lips are very important. And you would think it's for kissing, but it's really not for kissing, it's for speaking. You need to modulate your lips very carefully in order to be able to emit the correct sounds. So this map is very, very related to the language ability that humans exhibit. Okay, so we come to this area, and if I'm going to move my right arm, then I need to implant on the left side of the brain because the control is on the opposite side. So I go to the left side of the brain, I go to the arm part of the homunculus, and I implant an electrode. This is an electrode array, it's 10 by 10, so it's 100 electrodes. It's actually typical 96 because there is no electrode at the corners. It's about 4 millimeters on the side, so the distance between two adjacent electrodes is about 400 microns. And we record activity. We record n neurons, so n is the number of recorded neurons. It's of the order of 100 for this type of multi-electrode array, and is of the order of thousands for a new technology that has been used for the last couple of years, which is called neuropixels, which is really allowing us to record up to 5,000 neurons simultaneously, which is a lot. I'll show you data that in the 80s and 90s, we were only recording one neuron at a time. So I organize my data in this manner, and I am going to argue that this is typical of any data matrix. If you measure several degrees of freedom or several quantities at the same time, you put each one of them in a row. So this is, in this case, neuron 1, neuron 2, neuron n, where capital N is the total number of variables that you're measuring, in this case, the total number of neurons. And you measure at time t, and then you measure at time t plus delta t, and t plus 2 delta t, and you go on until you finish your trial or your experiment. So this data matrix has n rows, n is the number of degrees of freedom that I'm measuring, and has t plus 1 columns, where this is time or number of trials. So this structure is, this rectangular structure is the typical rectangular structure of any data matrix, and it's very rectangular because typically you measure a few degrees of freedom, so you have few rows, but you measure them for a longer time or for many trials, so you have many columns. So every column is a trial, remember that. And n is the number of recorded neurons.
So how do you insert those microchips? Does it have to be a surgical procedure, or does
it's a surgery. You have to remove a little bit of the bone, and then you implant them. And actually, it's interesting how you implant it. You implant them by pushing in. Actually, the tool that they use is a little bit like if you have seen people stapling a carpet, that they have these staplers that go boom, 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 like power staplers. So it's something a bit like that. It uses pneumatic air, actually, to press it against the, it doesn't go in, it stays in the top of the skull. And you need an opening for taking the wires out. So there have been some clinical trials where this has been done on humans, actually. And the problem with these electrodes is because they are metal, they are not biocompatible, really. So over time, you get scar tissues around some electrodes, so those electrodes stop recording, or the electrode goes down a little bit as a whole into the brain because the brain is soft in some sense, and so you start recording from other neurons that you were not recording before. So it's not very stable. We keep them in monkeys for 2 to 3 years, and we have kept them in humans for almost 6 months because we don't know the long-term consequences of having this metal implanted. There is a lot of work going on in creating biocompatible, polymer, conducting polymer materials that would be less invasive. You can do recordings with less invasive mediums like, imaging, but then you don't have the neuron by neuron resolution. These electrodes, you can capture one neuron at a time in each one of the wires. So it's a question of resolution versus invasiveness. Okay, so here I show you again what the population activity looks like. Again, I'm showing you of the order of 80 neurons in a given recording session. These are the targets, color-coded as before. Every row is a neuron. Every tick is a spike, and action potential is again about 20-something seconds, 20 seconds of recordings. And here, as time progresses, the monkey made a reach to the yellow target, and then a reach to the red target, and then a reach to the blue target. So the first question to ask is, by looking at the neural activity after target presentation, around the time of the go signal, would I be able to tell by looking at the neural activity to which target the monkey is moving? Is there enough information for me to read out the motor intent of the monkey just from looking at the activity in this in a given period of time? So what I do is I construct these boxes. They have a width delta t, which is about 300 milliseconds. And during these 300 milliseconds, I record the number of spikes. And I record the number of spikes for every possible neuron during that time period. And this is about 350 milliseconds after target presentation in each case. So I have per trial one data point in a space which I call the neural space, in which each axis represents the activity of a neuron. So I can construct a space where the axes are neuron 1, neuron 2, neuron 3, neuron 4. I define a firing rate by dividing the number of spikes that I recorded by 300 milliseconds. So I get spikes per second. And I define a firing rate. And I have 1 point per trial. And the question that I ask myself is, are these points organized in this neural space in such a way that I can make predictions as to which target was going to be the target for that particular trial? Well, I cannot show you an 80-dimensional space, but I can show you a 3-dimensional space. So I pick 3 neurons. I pick this neuron 78, which has kind of a normal firing rate. I pick neuron 46, which fires a lot. And then I pick neuron 70 because it fires very little. So remember, in this neural space, which is now 3-dimensional, every trial is represented by a point because I have the coordinates of that point. I know the activity of each one of those neurons. And now I can ask myself, are these points organized in any way? So what I do after I have all the points in that space, I color them. I color according to the target. And very unfortunately, this is what it looks like. So I have a population activity. I'm measuring n neurons. I have a firing rate vector, if you wish, a point in this n-dimensional space, which is the firing rate of each of the individual neurons. This is what we call the neural space. Here I'm showing you 3 dimensions, the 3 neurons that I mentioned, neuron 46, neuron 70, neuron 78, a point per trial. The firing rate is the number of new spikes that I saw within this box divided by the width of the box. So that's the firing rate, number of spikes divided by delta. And then I color coded, and boy, this doesn't tell me anything at all. There is no clustering. The points are intermingled. They are everywhere. So that doesn't seem like it doesn't seem like I'll be able to answer this question in any successful manner. So we went back and said, okay, what is known about these neurons? And what is known about these neurons comes from the 80s. At that time, we were doing single neuron recording, not we, but the lab of Georgopoulos. So now it's again the same task, the same centered out task. The hand is reaching in these 8 directions. Now every row represents not a different neuron, but a different trial. So there are many trials going up and many trials going to the left and to the right, down, and then the other four intermediate positions. And for this particular neuron, the firing rate was measured as a function of the kept track of as a function of the direction in which that particular reach was being performed. As you see, there is a lot of variability from trial to trial. Remember, it's a single neuron, and every row now represents a different trial. We'll go back to the population in a second. So there is a lot of variability, but at the same time, there is a lot of specificity. This neuron clearly fires more when you make movements to the left and either a little bit up or a little bit down. It fires kind of normally when you make a movement down, but when you make movements to the right, this neuron actually decreases its firing rate. So the Georgopoulos group decided to plot this average firing rate, again in hertz, number of neurons per number of spikes per unit time, as a function of the angle. There are only 8 angles here, 0 is to the right, 90 degrees is to the top of the figure. So you have 8 points here. And they thought it would be wise to fit these curves to a cosine curve. So I have a preferred direction, theta I, at which this curve has a maximum. So I measure the cosine of theta minus theta I. That will go between -1 and 1. I add a 1 to it, so now it goes between 0 and 2. I divide by 2. I take a half of it. So now this whole thing goes between 0 and 1. And I modulate the amplitude of that cosine by a parameter which is the amplitude of modulation, and maybe also by R, which is the extent of the reach. And then, of course, there is a background firing rate. So there are 3 parameters here: the background BI, theta I, which is the preferred direction of this neuron, and A sub I, which is the amplitude of modulation. So these 3 parameters within this cosine tuning model are sufficient to specify the firing rate of this neuron within this simple model of cosine tuning to movement direction. So I asked myself, imagine that these neurons in motor cortex were like a system of independent neurons. They don't talk to each other, which is clearly a very false assumption to make, a very extremely simplifying assumption to make. But what they do is they fire together because they are all seeing the direction of motion. So they fire according to their tuning curve. They all have a tuning curve. So if this was the case, what would I see at the population level? What would be my population of firing rates? So you can do that, and you can ask, what is the population of cosine tuned neurons do? So now my firing rate is a vector. It's firing rate of neuron 1, neuron 2, all the way up to neuron n. And it's just, a page of algebra. It's very simple to do. You get the vector of baseline firings. Then you get your 1/2 of R. You get A, which is your vector of modulation depths. And then you get a linear combination of 2 vectors, phi x and phi y. These are 2 n-dimensional vectors. Phi x is the amplitude of modulation projected onto the x-axis by the cosine of the preferred direction. And then this is projected onto the y-axis by the sine of the preferred direction. So if you go back here, you take A1 times the cosine of this angle, or A1 times the sine of this angle, and that gives you the first component. And you do that for every neuron. So now, as I vary theta, if I vary the direction of the reach continuously instead of 8 directions, what would I expect to see? Well, I expect to see a linear combination of these 2 vectors. These are 2 vectors in the n-dimensional space, and a linear combination of these 2 vectors will give me a ring. And if R is not fixed by there is a little bit of variability in the extent of the reach, then this ring will have a little bit of a depth. So although this lives in an n-dimensional space where I didn't even specify n could be the 100 neurons that I'm recording, or n could be all the neurons that are participating in this task, which in the case of a primate, human or non-human primate, is of the order of 1 million. Of the order of 1 million neurons in your primary motor cortex are being modulated in their firing rate when you execute this center outreaching task. So in this million-dimensional space, neural space, I actually expect to have this activity organized in 2 or 3 dimensions. So it should be possible to find those dimensions. It's just that it's not possible to find them by doing something stupid, like selecting 3 axes at random and pretending that this is the correct 3-dimensional space to look at, because then when I project into those 3 dimensions, I don't see any structure at all. So what I want to do is something a little bit smarter that will allow me to find this ring. And it turns out that you have the tools to do that. And this is where now it becomes a very general approach to how do you deal with data. And then I go back to show you what happens in the neural data. So your data is in the form of n-dimensional vectors. N is the number of variables that you measure, okay? So here, the data is the n-dimensional vector of firing rates associated with each reach. M is the number of trials. If I do M reaches, what I get is an n by m matrix. So X is a vector for a given reach, for a given trial, and X is this matrix where each one of these instantaneous measurements is one column vector. So it's an n by m matrix. So the first thing that we do, as always, we are doing to do principal component analysis or any kind of analysis, is we want to remove the mean. We want to go to a coordinate system where the data is centered at 0, not centered at the mean. So we compute the mean. Well, I don't really know the mean, but I can estimate it from the data. A given neuron is a row here. So I fix the row. I fix x, I, k. I is fixed. That's the neuron index. K is the column index. So k goes from 1 to m, and I divide by m. So I compute the mean along a row here. That's the mean, that's my estimated value of the mean firing rate of neuron I, and I subtract that mean from all the elements on that row. So now I have I have center data, 0 mean data. Once I have 0 mean data, I can estimate the covariance matrix. I can estimate the covariance matrix by doing x transpose and dividing by m minus 1. That's the way to get an unbiased estimator. I hope you have learned these things somewhere. And so the ij element
We
of
haven't,
this,
but it's in the notes. I didn't do it in the lecture.
Okay. So you divide by m here to get an unbiased estimator of the mean, but you divide by m minus 1, which is not what you would have guessed, to obtain an unbiased estimator of the covariance matrix. So this is how neuron I varies with respect to its mean, to estimated mean. This is how neuron J varies with respect to its estimated mean. And I correlate the fluctuations of neuron I to the fluctuations of neuron J by summing over all the trials, by summing over all the columns. So this is how I get the covariance matrix. Once I get the covariance matrix, I can diagonalize it. I diagonalize the covariance matrix, which I keep on the hat there because it's an estimator of the covariance matrix. And this estimated covariance matrix will yield eigenvectors and eigenvalues. The eigenvectors are the principal components, and the eigenvalues are the weights of those principal components. So principal component analysis is really a special case of something that you have learned, which is singular value decomposition. So let's go back to our data matrix X, which was an n by m matrix. The singular value decomposition is of the form U sigma V transpose. U is n by n. It's an orthonormal matrix. Every column of U is a vector. It's a vector in the neural space. Remember, the n dimensions correspond to the n neurons. And each one of these columns is a vector, is a direction in that space. They are normalized to be of length 1, so I only care in which direction they point. And if you look at 2 different columns, they will be perpendicular to each other. That makes U an orthonormal matrix. V is also an orthonormal matrix, but is in the m-dimensional space. It's an m by m matrix, and it provides a basis not for the neural space, but for the space of samples, for the time evolution, if you wish, or for the trial by trial. And the V's are the columns of this matrix, which is also orthonormal. And here I put it as a row because I'm supposed to put V transpose here. So U is n by n, where n is the number of neurons. V is m by m, where m is the number of samples. And in the middle, I have a matrix sigma that has to be n by m. And the matrix sigma has an n by n block, which is diagonal, has diagonal elements and then zeros outside of the diagonal. But it has to have the right size. So the remaining block, which is n minus m, has just zeros. And if you think of multiplying sigma by v, these zeros here will just tell you that the first n eigenvectors of v are important, but that the remaining m minus n I actually never seen. You know, they are what is called a null subspace of this matrix from the point of view of this mapping, if you wish, from the m-dimensional space to the n-dimensional space. So this is a singular value decomposition, and you've seen that. So now, from the singular value decomposition, and this is the only bit of algebra that I'm going to do. Actually, no, there is a little bit of algebra in the last half hour, but this is one of the two big bits of algebra that we're going to do. We are going to derive principal component analysis as a special case, in some sense, of singular value decomposition, or as a derivative calculation to singular value decomposition. So we have decomposed our matrix X in this way, U sigma V transpose. So we have to construct X transpose to construct the covariance matrix. So what is X transpose? Well, X transpose, I have X, which is U sigma V transpose. And then I have to transpose these. So when I transpose, things get permuted. So I get V transpose, which is V, and the sigma transpose, and then U transpose. So here I have the product of V transpose V. V is an orthonormal matrix. Therefore, V transpose V is the identity. I can get rid of it. So I get U, I get sigma transpose, and I get UT. So now what I have done, sorry about that, is I have diagonalized my covariance matrix, which is X transpose. The covariance matrix, which was 1 minus m minus 1, 1 divided by m minus 1 times X transpose, can be written as a form of U lambda U transpose, where lambda is the sigma transpose appropriately normalized by n minus 1. And you can check, this is something that you can do yourself, is to take a matrix sigma of this type, and you construct sigma transpose. What you will get is an n by n matrix that is diagonal, and its diagonal elements are the square of the diagonal elements that appear in sigma. So lambda, the lambdas are the square of the sigmas. And you know in the back of your head, the lambdas are going to be variances. So the sigmas are essentially like standard deviations, and this is taking the square of the standard deviation. It's not really the standard deviation because I still have to divide it by n minus 1 to get the standard deviation. And then this is going to be a variance. So the lambdas are the squares of the sigmas in some sense. This is what you need to remember, heuristically speaking. So then we are done with our principal component analysis. Lambda is this matrix 1 over m minus 1, sigma transpose. Lambda now is a square matrix, n by n, and it is diagonal, has zeros outside the diagonal, and the elements of the diagonal, which are the eigenvalues, are traditionally ordered. They don't have to be, but they are. Algorithms give them order to use. So lambda 1 is the biggest one, and then lambda 2 all the way to lambda n. Okay, so far all that I have done is show you how principal component analysis is a special case of singular value decomposition. But what I wanted to do is do some kind of smart dimensionality reduction, right? I wanted to find this kind of three-dimensional structure, which is sitting somewhere in a very high-dimensional, n-dimensional space. So how do I use singular value decomposition or principal component analysis, more specifically, for dimensionality reduction? Well, I have this U matrix, which is an n by n matrix, whose columns are the eigenvectors of the covariance matrix. And what I do is since these are also ordered, the U1 goes with lambda 1, U2 goes to lambda 2, and every eigenvector goes with the corresponding eigenvalue, I am only going to keep the k leading eigenvalues or the k leading eigenvectors. So I call this matrix U tilde. It's again a rectangular matrix. It's n by k. And I'm going to construct a covariance matrix which uses U tilde instead of U. So it's U tilde lambda U tilde transpose. So before I have a way to write the covariance matrix, the full covariance matrix in terms of the eigenvalues and the eigenvectors. So this is U transpose. That gives me an n by n matrix. Is weighted by the corresponding eigenvalue, and the sum from this from 1 to n is what is called sometimes the spectral decomposition or the eigenvector decomposition of the covariance matrix. But what we're doing is, yeah?
Just a quick question. I was wondering how you decide like how big K is.
It's an excellent question, and that I will get there. I mean, you can choose K, and then this is a very well-defined procedure, but the whole trick is how do you choose K? How do you know what should be that element? In this case, for my neural data, I gave you a hint. I used a previous result to say, I'm going to look for 3 dimensions. But in general, how would you choose K? And I'll give you some hints about that. It's not a mathematical theorem how to choose K, I should say. You know, choosing K is always a little bit of trial and error, but it's an excellent question. So I replace my covariance matrix by a K dimensional. I only sum over the K leading eigenvector. So now what I'm doing is, before I was in this n dimensional space, right? And the columns of U gave me a basis for that space, where n vectors, all of them of length 1, all of them perpendicular to each other, and this span, any point in that space can be written as a linear combination of these eigenvectors, of these principal components. Each one of them is not a neuron, it's a direction in neural space. An eigenvector points into a direction which is not neuron 1, neuron 2, or neuron 3. It's not the original axis. It's a particular pattern. It's a particular combination of neural activity. But now what I'm doing is, instead of keeping all n eigenvectors, I'm keeping only k. So that subtends a subspace of dimension k. For instance, if I only keep 2 eigenvectors, they will subtend the plane. And that means that only I'm projecting all the points onto that plane. And the components of the points outside of that plane are being ignored because I'm not keeping those directions at all, right? But instead of a plane subtended by neuron 46 and neuron 78, it's a plane subtended by the first and second principal components, by the first and second eigenvectors, which correspond to the first and second eigenvalues, which is the largest eigenvalue. So I'm keeping the directions that have most variance in terms of how much the points are spread in those dimensions. And I'm throwing away the directions that have less variance. And that's exactly what we're doing. So we are doing a dimensionality reduction by going from a formulation in which we just change the basis, and we have a complete basis. We have all n eigenvectors that span the space, into an approximation, in some sense, in which I only keep k. So for instance, these are my points spread in 3 dimensions. So I have a principal component 1, which is a direction. It's very important that first of all, it's very important that I subtracted the mean. So my coordinate system now is centered at the center of mass of these points. The points have mean 0 about the new coordinate system. Now, the orientation of the axes in this new coordinate system are not the original axes, are not x, y, and z. They are PC1, which is the direction along which I have the most variance of the data, PC2, which is the direction that follows in amount of variance, and PC3, which is a little bit of variance, which means there is very small fluctuations out of the plane subtended by PC1 and PC2. So now if I look at the 2-dimensional plane subtended by PC1 and PC2 and project all the points there, what I have lost is very little. Because what I have lost is the fluctuations in directions that are outside of that plane, but they are directions along which the variance is very small. Therefore, the deviations relative to that plane are very small. So this is the rationale for doing a cutoff that is controlled by an arbitrary k, but a k that takes into account an order which is given by the eigenvalues. So it's a variance-guided dimensionality reduction. I'm going to keep the most important dimensions. And this is the great miracle that happens. So this is a totally useless plot that I had shown you before, where I pick 3 axes. And now, oh, I should have said that we call each one of these directions a neural mode, because each one of these directions, direction PC1, is, as I said, a given pattern of neural activity. For instance, it might say, when neuron 1 fires, neuron 2 should fire twice as much, neuron 3 should not fire at all, neuron 4 should fire negatively, means it's firing less than it's firing, less than its mean, and so on and so forth. So it's a given pattern of, it's like a mode in an elastic system, or in a quantum mechanical system, right? So here are my 3 leading eigenvectors. You can think of them as PC1, PC2, PC3, neural mode 1, neural mode 2, neural mode 3. So now I do the same exercise. I put 1 point per trial, and after I have put all the points, I color them. And this is, this is a relatively new result. We've only known this for 10 years. And it's a fantastic result because there are 2 things here. First of all, it's clustered. So there is an organization according to target. But moreover, and this was really totally unexpected, the clusters obey the spatial organization of the targets. I go from orange to mustard to yellow to green, like from orange to mustard to yellow to green. And then the blues and then the red, the aquas and the blues and then the red. So there is a neural representation in this very high dimensional neural space that is capturing the spatial organization of the targets in the outside world. You have learned this task, you've done this task many times, you have a neural representation of the task, and clearly it's very useful for this neural representation of the task to be able to have this neural representation of the external world in which this task is going to be performed. So now this is insofar is purely static. I'm not talking to you about the trajectories to go to the target and come back. This is 1 point per trial. So this is a little bit like if you play the piano, not having the sequence representation yet, but having a representation of harmonics, having a representation of targets that you tend to play together. You tend to play this accord versus that accord versus, put your fingers in this particular place versus fingers in that particular place. So this was a really profound discovery because it really reinforced this idea that there are these low dimensional representations in the brain. So we found them in motor cortex, but then after we found them in motor cortex, people started finding them in many other areas of the cortex, prefrontal cortex, sensory cortices, having to do with vision and audition and taste and tactile representations. So this is a little bit what is happening. I'm going to play this to you again. So I have a very large neural network, as I said, of order 10 to the 6, if I'm thinking of this area of your primary motor cortex that is being modulated or participating in some sense when you do these centered outreaches. I'm subsampling terribly. I'm putting an electrode that records only neuron 1, neuron 2, and neuron 3 out of the many neurons that are there. And I see these raster plots. This is the spikes for neuron 1, the spikes for neuron 2, the spikes for neuron 3 as a function of time. But now, if I take my interval of size delta t, and I follow the point as a function of time, what I see is that the dynamics is actually constrained to a low-dimensional subspace. Not only the representation of the targets, but the dynamics itself during the motion is constrained to this low-dimensional subspace. So it's enough to study population activity in these low-dimensional spaces that we call manifolds, neural manifolds. And this is a very prevalent phenomenon in the brain. So there is a lot of investigation going on right now, a lot of research going on right now. First of all is what causes it. In principle, you will have this very high dimensional nonlinear dynamical system. The neurons are nonlinear, the synapses, the connections between them are nonlinear. So you have this nonlinear dynamical system evolving in a 10 to the 6 dimensional space, in a million dimensions, and yet its activity is confined to a few dimensions. So you have to ask yourself, how is this happening? And the idea is that it happens because when you learn the task, the connectivity of the network changes, and that the low dimensional dynamics are reflecting the connectivity of the network. And then the other thing is, okay, once that you know that the activity is confined to a low dimensional space, it becomes easier to study. So I have to be able to characterize a low dimensional space and then characterize the dynamics on the low dimensional space. And all this is very, very active areas of research right now.
So could I ask?
Please.
So this seems very convincing, but I just want to make sure that in the data we're not selecting sort of, I guess, pre-selecting what we want to look for, right? So we're choosing modes with a high variance, right? And so
exactly, I'm selecting according to variance, and it's a very good question. Is that a good criterion, right? Mm-hmm. Is that should I select according to variance or not?
And what are we missing through that? Yeah, if any.
Start again.
And what are we missing when we choose based on that criterion, right?
It's a very, very good question. We're missing clearly all the low variance dimensions, and the assumption is, but it's an assumption that needs to be checked, is that the low variance directions correspond to fluctuations in and out of this hyperplane, in and out of this manifold, and that those directions correspond to noise. The system, neurons are very noisy, the system is very noisy. So the idea is that high variance is information, low variance is noise, so it's very fine to neglect the low variance. But actually this needs to be checked, because imagine that I have one direction out of this manifold, but imagine that along that direction, the fluctuation is always positive, and it's positive only when I'm going to target number 3. So that would be a very informative fluctuation. It would be low variance, but it would be very reproducible and very informative. So the idea of saying low variance is not informative and it's okay to neglect it is an idea that needs to be challenged and needs to be verified in each case. But the dogma, not the dogma, but the hypothesis behind this method for dimensionality reduction, which is variance-based, you're absolutely correct, is that high variance is the informative directions in the space, and low variance are the noisy fluctuations, and therefore it's okay to neglect them. Now, for instance, I can play my game, and this is a way to select K, which was the question that, I'm sorry, I forget your name, asked before. I don't see your gallery, sorry.
His name is
One
Andrew.
of you, say it again?
Andrew. His name is Andrew.
Andrew, thank you. Andrew asked before, how do I choose K? And so a question would be, for instance, your name is Andrew. No, I was asked, somebody asked before.
The other one was Adam.
Yeah,
Adam. Thank you.
Yeah.
So Adam asked earlier, how do I choose K? So one way to choose K is, okay, I say, okay, I'll pick K equals 1, and I will try to predict to which one of the 8 targets I'm going. And then I choose K equals 2, and then I choose K equals 3. And then at some point, my ability to predict will saturate. I can increase in K, but that means that what I'm adding is noise. It's not informative. So it's a way to check a posteriori that it was okay. Let's say that after 3, I get most of it, and then 4, 5, 6, I get a bit of improvement, and then no improvement after that in my ability to predict to which one of the 8 targets I'm going. Okay, so it's noise from the point of view of this task, which was it clearly requires knowing to which one of the 8 targets you're going to move. So, but these are a posteriori checks but Andrew, you're absolutely correct. This is an hypothesis or an assumption that needs to be checked. But this brings me into the topic of Adam's question, which is, what is the way of choosing K? So one way of choosing K is to look at the eigenvalues. And if the eigenvalues decay, like, first eigenvalue, second eigenvalue, and then all the eigenvalues are the same, that would give me a knee in the eigenvalue distribution that would tell me how to choose K. Unfortunately, when I plot the PCA eigenvalues for this task, the eigenvalues corresponding to the principal component analysis, is a very soft decay. And, I could keep 1, I could keep 2, I could keep 3, I could keep 4. I mean, there doesn't seem to be a good reason for choosing a particular value of K. I want to show you a different method for dimensionality reduction that is nonlinear, that is called Isomap. And what I want you to keep in mind as a motivation for learning about Isomap is that when I plot the Isomap eigenvalues, I get the blue eigenvalues. So eigenvalue 1 is important, eigenvalue 2 is important, and all these eigenvalues are almost constant, which means that I have like a coin. I have 2 dimensions that are important, and then in all the other directions in this space, I have a thickness, a variance, which is almost the same. This is the size of the noise in some sense, right? Is the amplitude of the noise, is the strength, is the amplitude of the fluctuations around, in and out of that manifold, in and out of that hyperplane. Well, in the case of Isomap, it's a real manifold because it's not flat. I'll show you how that works. So we always start with linear methods, but we always try to do at least one nonlinear check because it gives us intuition about how to choose K and about whether choosing by variance was a good idea or not. So this is how Isomap works. Isomap is a method for nonlinear dimensionality reduction. So far, I showed you PCA, which is we started in neuron 1, neuron 2, neuron 3, and we created a 2-dimensional hyperplane that then we can look at it as in 2 dimensions, X and Y, where the directions in the XY plane, each one of them is not a neuron, but a neural mode. It's a direction in neural space. Isomap is doing something different. The end effect is the same, but it's not putting a hyperplane through the activity, but it's trying to put a nonlinear surface through the activity. So it's a nonlinear method for dimensionality reduction. So although this is a hyperplane, this is what is called a manifold or a curved manifold. You can think of a hyperplane as a flat manifold. So manifold is kind of the generic name. So I talk a lot these days when I talk to my colleagues about neural manifolds and whether they are flat or whether they are curved. So where does Isomap come from? It comes from a very useful technique to know, which is, again, and you'll see surprisingly, all based, is a linear method for extracting nonlinear manifolds. So the idea is I want to represent points in a low-dimensional space in such a way that the Euclidean distance between these points reproduce as well as possible an empirical matrix of distances or dissimilarities. So I have these objects A, B, C, D, and I have a similarity matrix. So A is similar to itself, B has 0 distance to itself, C has 0 distance to itself, and D has 0 distance to itself. But there is a distance from A to B, C, and D, and so on and so forth. It's a symmetric matrix. It's a non-symmetric. It's a symmetric matrix. It's a matrix of distances. And I want to put these points as points in a 2-dimensional Euclidean space, in this case 2-dimensional, such that the Euclidean distances between them reproduce as well as possible this matrix of distances or similarities. So this is actually very cute problem. It arises because at some point, the kind of polls or study groups or taste groups that people who want to do marketing develop. So people went around Scotland asking people to sample whiskies and tell them where these whiskies were very similar to each other or very dissimilar to each other. So they created this totally subjective matrix of similarities and dissimilarities, and then they averaged over all the people they had consulted, and they got these kind of matrices. So this is 4 whiskies, and this is how people judge them to be similar or dissimilar to each other, and this is the embedding of these 4 whiskies as points in a 2-dimensional space, so that the distances, Euclidean distances between them reproduces as well as possible the matrix of similarities or dissimilarities. So how do we do multidimensional scaling? And this is the second part where we'll do a little bit of algebra. But again, this is in the books. It's a very well-known technique. Again, the same form for the data. Every data point is an n-dimensional vector. Here, the data would be the n-dimensional vector of firing rates associated with each reach. If I have m reaches, I get my data matrix, which is again our old friend, the m by m data matrix. Now let's say I don't give you the data matrix, but I give you a matrix of distances. Now, I have m such points here in the n-dimensional space. M is my number of trials, or my number of samples. So I can create if I create a distance with all the pairwise possible distances as an m by m matrix of distances, it will have zeros in the diagonal, it will be symmetric. And the question is, if I give you the matrix of square distances, can you reconstruct the matrix X? Can you tell me which points were they? Well, clearly, except for rotations, right? And except for the center of mass, but we take that away by going to the zero center of mass, by subtracting the mean. So we subtract the mean. So except for rotations, can we know where the points that have these distances? And it turns out that there is a solution, and it's a linear algebra solution if the distances were Euclidean. If the distances were Euclidean, then the distance squared between point I and point j is xi minus xj squared. So xi minus xj is a column vector, its transpose is a row vector, the scalar product between them gives me the distance squared. Now, the distance squared can always be written in terms of the distance is the absolute value of one plus the absolute value of the other minus the scalar product. So the scalar product can be solved for given the distances if we happen to know the norm of these vectors. And you're going to say, but I don't know the norm of these vectors. But it turns out that this is a beautiful trick. This takes, again, about maybe 1.5 pages of algebra, that you can actually write the matrix of scalar products, the X transpose X matrix of scalar products between the data points, given the S matrix, which is just a matrix of distances, by operating on it with a matrix that actually gets rid of these norms, because we don't know them. We don't know what are the norms of these vectors in the Euclidean space, because we don't know the vectors in the Euclidean space. We only know the distance between them. So this matrix is the identity in m-dimensional space, and this is a vector that has all ones. So this is the outer product, not the inner product. It's not E transpose E, but E transpose. So this is the column vector, and this is the row vector. So this gives me a matrix which is filled by doing this outer product between two vectors that have all ones as entries, and there is a 1/m normalization. So this is a matrix. Multiplying by a matrix, both right and left, is a linear algebra operation. This is called a centering matrix. So it turns out that if you took your matrix of distances and sandwiched it between j and j, and took a half of it and with a minus sign, you would get X transpose X. And this can be verified. This is just identities in the case where the distances were Euclidean. Once you have X transpose X, you can get X, because X transpose X is essentially the square of X. How do you get X? Well, you get X transpose X. You do your decomposition. You do your singular value decomposition. In this case, U and V are going to be the same, and you can verify that. And so if X transpose X is of this form, then X is of this form. X is the square root of lambda times U transpose. And there's no problem taking the square root, because you remember this is a squared essentially. So the elements of lambda were variances. The elements of lambda were the squares of the elements of our original sigma. Okay, so what do we know here? What we know is that if the distance matrix to this calculation is applied is based on Euclidean distances, this process would allow us to recover the data matrix X from the distance matrix S. That's totally obvious. It's identities. I could do dimensionality reduction by going to this equation, and instead of keeping all m eigenvalues, just keeping all k eigenvalues. And that means only using k eigenvectors to reconstruct x. So now my points x would lie in a k-dimensional subspace. And you can prove that in this case, this translocation is equivalent to PCA, except that it's working on PCA is smarter. PCA works on x transpose, which is n by n. This works on x transpose x, which is m by m. And remember that m is much larger than n, typically. So this is a stupid way of doing PCA because it's looking at the other covariance matrix, the covariance matrix that works in sample space, but it has the same eigenvalue spectrum. Okay, so all this is linear so far. I was in an Euclidean space, everything was flat and linear. I just showed you that given the distances, you can extract the data. Good. Now I give you square distances, which are totally arbitrary. I apply to an arbitrary matrix S of square distances that were obtained in a very phenomenological manner. So the method still implies getting this matrix Y through the centering operation. So you give me the S, I sandwich between J and J, I divide it by 2, I put a minus sign, and I call Y. I do a diagonalization of Y in the form of U lambda U transpose, and identify X as 1 the square root of lambda times UT. And so this procedure, what is giving me is a matrix X which is as close as possible as the matrix Y, which was obtained from the real Euclidean. So this is now an Euclidean representation of the data that is trying to mimic as well as possible a representation of the data that came from square distances that were not necessarily Euclidean. And this is the beautiful idea of Isomap, and this is a very famous science paper by Josh Tenenbaum. And the whole trick is how to compute the matrix of distances. So he said, this very complicated manifold. I have a point here and a point here. This is the Euclidean distance between these 2 points. But I don't want the Euclidean distance. I want the distance by walking on the manifold. So how am I going to do it? He said, okay, let me take a little epsilon ball around this point, and the points that are within the epsilon ball, I'm going to compute those short distances as if they were Euclidean. I'm going to make an assumption that locally the space is flat. So here I am, and I'm going to look at my 10 neighbors or 20 neighbors, and I'm going to compute the distance from me to them and from them to me in an Euclidean manner. So I begin to fill the matrix of distances with all the short distances. But now I have these 2 points which are not neighbors of each other. So how do I compute the distance between them? Well, I walk on the manifold by taking short steps whose length I know because I made a local assumption of flatness, a local Euclidean assumption. So although this would be the true blue distance, what I go is to say, go from this point to this point, to this point, to this point. I don't know if you see the walk, but the walk is going through points. And all the steps in the walk are short, so I know their length. So I can add the total length. Now, of course, there are many ways of going from this point to this point by walking in the manifold. I'm going to choose the shortest one of them. And that's the only complication in the algorithm, is to be able to do all these possible walks in the manifold, choose the geodesic in the manifold, the shortest distance. When you apply it to this kind of data, it provides you with this kind of representation, of 2-dimensional representation. So it unwraps, it gets rid of the curvature of the surface. And this is what we did with our data, and this is what we got for the eigenvalue representation. So the eigenvalue representation now clearly told us that our data was 2-dimensional. Remember that I showed you, actually, now I'm going to go way back, I show you a 3-dimensional representation of the data here. But actually, Isomap said, there is a 2-dimensional surface there. It's just that it might be, maybe it's not flat. It's a little bit wiggly. This is why you needed 3 dimensions to see it. But if you allow the manifold to be curved, you can put all this data actually in 2 dimensions. You do not need 3. And how do I know that? I know that because of the eigenvalue distribution that tells me that 3, 4, 5, this is all the amplitude of the noise. And these are the only two eigenvalues that are really relevant. So we did that. We did the projection guided by Isomap in a two-dimensional space. And now you can see again the clustering, and you can see again the organization where you go from orange, red, yellow, and you're preserving the topology of the space. And I'm sorry, this is a wrong representation. I need to change that. How do we know that the manifold is curved? Well, we look at the distances. We look at the geodesic distances that we had obtained by walking on the manifold, and we compare them to Euclidean distances. And the geodesic distances were bigger, which means that we really, by walking on the manifold, we are taking a longer path than by going, as the fly flies from point A to point B. So clearly we're being forced to walk along a curved surface. And I'm going to end there, but I want to give you a little bit more intuition about this kind of flat versus curved manifolds. So I'll talk to you about neural modes. So I started with a space where the axes were neuron 1, neuron 2, neuron 3. And I said, well, maybe the dynamics is confined to a hyperplane. And if it's confined to a hyperplane, that's the case of a flat manifold. Then I can construct a basis. There's these two eigenvectors, U1 and U2, that are a global basis for the hyperplane, that tell me how the hyperplane is actually, every point in the hyperplane is actually a linear combination of these two eigenvectors, of these two principal components. But what if the manifold is curved? The manifold is actually like this. Then locally, in every point, I have a basis, U1 and U2. But then as I move along the manifold, this basis is changing. It's not a global manifold. So we somehow want to be able to distinguish between these manifolds that can be curved and the flat manifolds that are much more appealing to us. We have a much better intuition. So I learned this description. We had a workshop on this at the Computational Neuroscience meeting on these manifolds for neural computation, and two colleagues of mine, Srdan Stojic, who is at the Colnorman in Paris, and Ila Fiete, who is at MIT, presented a very nice intuition, and that's the last thing I want to show you, no algebra at all. So imagine that I have a shoelace, okay? What is the intrinsic dimension of this object? The intrinsic dimension of this object is 1. It's a ring. I can go around it. And it's a one-dimensional representation, really. But if you want to see it in space, you have to lay it flat on a surface. So you need at least 2 Euclidean dimensions. You need at least 2 flat dimensions to see it. So for this object, the top one, the intrinsic dimension is 1, but I need at least 2 for an embedding dimension, where the embedding dimension is, you could think of it, what is the smallest dimension of the flat manifold on which I can embed this intrinsically non-flat object, okay? But now I take this object and I try to pull, I try to distort it. It was on the plane, but now I take a little piece and I pull it up, okay? So now it's no longer on a plane. So now suddenly I need 3 Euclidean dimensions to embed it. And now imagine that this object is sitting in a 100-dimensional space. There are many dimensions in which I could distort it. There are many directions in which I could introduce these little wiggles. And as I put more and more wiggles along different dimensions, my embedding dimension, the minimal number of flat eigenvectors that I need to describe a hyperplane that will contain it will increase. However, its intrinsic dimension is always 1. So this is why we always do first a flat analysis, and we ask ourselves, what is the smallest number of eigenvectors that define a hyperplane that allows me to contain this object? But then we should allow for the possibility that the object itself is actually a nonlinear object, a non-flat object, that is actually much lower dimension than our flat dimension, but is embedded in this flat space that we found. So if PCA tells me that you should keep 10 dimensions and should always allow for the fact that maybe there is a 4 or 5 dimensional nonlinear manifold embedded in this 10-dimensional space. This is another example where if I'm constrained to walk on the surface of the sphere, my intrinsic dimension is 2, but if I want to embed it in a flat space, I need at least 3 dimensions, right? And again, you can imagine taking this sphere in your mind and start adding little blobs of it that go into this, 12, 10, or third dimensional space of string theories. And then you can see how you need a flat space that is higher dimension is because of the wiggles of the strings, right? So try to always keep in mind the idea. So on one hand, this is a plug for linear methods. Always do your principal component analysis is very well justified. As Andrew said, always check a posteriori that the directions that you have thrown away because the world of variance are non-informative. And non-informative is depending on what this data is about. The data is supposed to contain information about something. So check whether you have captured most of the information about that something by keeping the k dimensions that you decided to keep, and that the rest is really noisy. And that also allows you to choose k. But the best way to choose k is really if you get an eigenvalue distribution like this, because then it's clear that you should use that by the time you get to 3, you have too many, and that 2 should be enough. But that often requires using a nonlinear method. And I had found Isomap to be particularly, there are many nonlinear methods, but I like Isomap mostly because in some limit, Isomap becomes PCA. If I increase the size of my neighborhood and I compute all the distances as Euclidean, Isomap becomes a stupid way of doing PCA, because I'm doing it on X transpose X instead of doing it on X transpose. So I'm diagonalizing an M by M matrix, which is very big, instead of diagonalizing an N by N matrix, which is quite smaller. But it is PCA. It's PCA done in a silly way, but it is PCA. You will get the same eigenvectors and the same eigenvalues, whether you diagonalize X transpose or X transpose X. You can check that actually, that you get the same eigenvalues and eigenvectors. It's very nice. So the same eigenvalues, not the same eigenvectors, because one set of eigenvectors is in n dimensions and the other set is in m dimensions. You get the same eigenvalues. Sorry, I misspoke. So always do PCA first. If you get an eigenvalue distribution that is very gradual, like the red points here, that doesn't really indicate that you should do any kind of dimensionality reduction, then look for a nonlinear method. And Isomap is a preferred one of mine. There are many nonlinear methods for dimensionality reduction. But it's interesting how the whole basis of Isomap is based on multidimensional scaling, which is again a linear algebra method. And the only trick about Isomap, the only trick where you really bring in the nonlinearity, was this idea of Tannenbaum and his collaborators of compute distances by walking in the manifold. That's the only place where you really take the nonlinearity into account. Local linearity, but global nonlinearity. And again, I use neural data as an illustration for how this works, because this is what I do. But this is a very general approach to doing data analysis, and it's based on some of the tools that you have already learned. So I'll end there.